Good morning, doctor and everyone. We are from Group 5, which consists of me, Chong Chien, and Kelly Eng. And our topic for today would be dermatophytosis under Microsporum gallinae. So for introduction, avian dermatophytosis, which is also known as FAVUS, is an uncommon superficial fungal infection seen in poultry. It primarily caused by the dermatophyte fungus Microsporum gallinae. And Microsporum gallinae is common in gallinaceous birds and other poultry, such as ducks and pigeons, and also has been shown to cause disease in mammals, including dogs, cats, mice, squirrels, cattle, and monkeys. It is also a member of the zoophilic fungi, which are a public health concern because they can cause severe acute skin inflammation in humans, which can expose to infected animals. So for the types of samples to be taken when dealing with a suspected case of Microsporum gallinae infection, the type of sample that we can collect would be crust, broken feathers from lesions, and also debris from the feather follicles. Lesions are typically can be found as the picture shown at the comb, around the eyes, and also can be seen around the neck. And this area should be carefully examined and samples should be collected from the edge of the active lesions where the fungal activity is most intense. Uh, Next, the sample techniques. One of the most common sample techniques would be skin scrapings, where usually it is conducted under clean conditions, and we should collect or obtain the skin or lesions with a sterile blade to prevent any contamination. Next, after collecting the sample, we should place it into a dry container such as petri dish, folded paper envelopes, or a sterile plastic bag. And if there's feathers that we can collect it can be clipped or pulled with sterile forceps. And for the transportation, it should be transported at a room temperature to avoid moisture. Next, for the direct microscopic examination, the first step is we should use a small portion of skin scrapings or feather debris and place it on a glass slide. And we can use the 10% KOH, which helps to digest keratin for better visualization, and mix it with the skin scraping and cover it with a cover glass. And then we can do observation under the microscope for any septate hyphae and arthroconidia. So the picture shows the morphology of Microsporum gallinae under the observation of the microscope, and we can see macroconidia, which are the most prominent structures. It is in a spindle shape and typically has 3 to 6 septae. And microconidia, it can be seen around the macroconidia, which are small and unicellular, and also hyphae can also be seen in this picture. So for the fungal culture, the type of media that we can use for Microsporum gallinae would be Sabrodextrus agar, the SDA agar, dermatophyte test medium, DTM, mycocell agar, and also potato dextrose agar, PDA. So how to breed? We can use a sterile or loop to press the sample into the surface of agar and inoculate in multi areas, which is usually in 3 dots or focus on 1 dot. For the time and temperature for the culture of the fungal, the most common would be 10 to 20 days and in a temperature of 25 to 30 Celsius. So this is an example of the fungal culture of the Microsporum gallinae. The left side of the slide is an example of the SDA agar, which is the Sabrodextrus agar, or the right side is the potato dextrose agar, which is the PDA agar, which is being cultured at 25 Celsius for 21 days. The morphology of the fungal under the SDA agar can be seen white and cottony with slight red pigment diffused into the medium, while for the right side for the PDA agar can be seen a pinkish gray with a felt-like texture on the surface, and also can be seen a slight strawberry red pigment diffused into the media.
So for the microscopic examination of the native culture, the first thing we have to make is making the native preparation. A common method for the obtaining sample is using tape preparation, and another one is direct slide preparation. It can be made by scraping the affected skin or lesion to collect the scales or hairs. So for the dye used, the most common one is the lactophenol cotton blue. And other than that, which is not that common one, is the potassium hydroxide, which is KOH. And this is the observable image for the microscopic examination. Okay, next. The procedure for making slide culture is firstly, we have to sterile the petri dish first. And second, we pour a thin layer of the solid agar, which actually is a liquid form that time. And after that, the fungal culture is then inoculate to the surface of the agar when the agar is cooled down and become a solid. And the slide is placed flat on the agar in the petri dish. After that, the dish is then sealed and incubated at a temperature for 25 to 30 Celsius for 3 to 5 days. And the fungal structure is ready to stain. Next. For the staining technique and microscopic image, the most common one, which is the lactophenol cotton blue, LPCB, and the not that common one, potassium hydroxide, got a slightly different way. For the LPCB, a small drop of LPCB is added to the fungal growth on the slide and followed by the application of the cover slip. Other than that, a drop of the 10 to 20% of KOH is added and the cover slip is placed on the top of it. Next. So for the main features of the microconidea, M. gallinae, it is large microconidea, smooth wall microconidea, multicell microconidea, and spindle-shaped microconidea. Next. So conclusion, both direct microscopic examination and slide culture are useful methods for identifying M. gallinae. The microconidea, which are large multiple cell and spindle-shaped microconidea, are the most distinctive feature for M. gallinae, and the presence of hyphae microconidea and occasionally chlamydospore further supports the identification of these dermatophytes. When these features are observed together, the diagnosis of M. gallinae is typically confirmed. Next. Okay, that's all from us. Thank you.